Very different from the thread-legged one that we've been seeing so many of today.
Copy that. Video. When you're happy, come wide. We got lasers on it real quick.
Yeah, copy.
Bob Carney suggests perhaps the genus Glyphocrangon.
Okay, yeah, I see that now.
And then there was a fish. Is this still the same fish, Washlead?
Yes.
Copy.
And Tammy Frank says the same thing.
Yeah, I saw they're in agreement, so...
Alright, video, come on in. And you can kill lasers, co-pilot.
I think this is, yeah, this is a cusk eel, and I can't remember if we've seen this particular one earlier in today's dive, which is why I wanted to take a look at it. Nice and very obvious lateral line there, which is how they sense water movement. Okay, that's probably good if video is happy with that.
All right, happy video.
Good luck, gentlemen.
Actually, do you want to switch over here? Copy. I'm okay, thanks. Alright, Nav, what's ship status? Should we put in a move?
Yeah, ship is currently stationary. We have reached waypoint 2, and it looks like the slope should start heading over to the west. So if you could line up, probably about like 305 is what IPAC's showing.
Okay, I will turn my head to 305.
Alright, I'm looking.
Sonar agrees.
Okay.
Yeah, 305 looks good to me.
305, I'll go another 20 meters.
Copy.
Bridge is ROV now.
He's a Lars. Sorry, he's stretched out. You want to just make it 30?
Copy that. Could we have a ship move, please? 30 meters at 305 degrees, speed 0.2.
See anything in that
Good
video?
copy, Bridge. Thank you.
More?
Yeah, if we can zoom in, that'd be awesome.
Just
one burrow full of anemone, another one full of sargassum and brittle stars. And it looks like the brittle star, if that's what it is trying to make a hasty retreat back into the burrow.
Alright, are you happy with that?
That's good, thank you.
Great. Video, if you're happy, come wide. And we're just going to do a quick shift of seats.
I wonder if those Glyphocrangon might be the constructors of some of these burrows. I'm not sure if they're burrowing shrimp or not, but let's see if I can find out.
Yeah, maybe Bob and or Tammy can chime in on whether or not Glyphocrangon could potentially be the builders of the numerous burrows we're seeing out here.
Welcome, pilot. Just for your reference, you have a sample in the starboard inner box. It's a sea pen. It's not expected to swim out if you open it.
Copy that. Good to know. Good to know. Doesn't make worm-like movements.
Yeah, it's a negative on being able to spring.
Only
Out of the
retracts,
box.
doesn't
Only
spring
retracts.
or swim or jump
No
or anything else.
extension
Copy
whatsoever.
that. I'm reading everybody.
But what you may have not have heard is we do have a ship move in at 305 for 30 meters. So
But-
we have reached
Okay.
waypoint 2, and now we're heading more towards the west to continue following the ridge up.
Okay. Sounds great. Just explore. Watch leads. If you see anything, just yell it out.
Only one acting watch lead
watch
at the
lead.
moment, so
Excuse me,
I'll let you
yes.
know.
Yes, watch lead.
But can we have you guys introduce yourselves? It's been a while, and you were just a partial switch a while ago, and I forgot to ask you to do so.
Sure. I'll go ahead and start. Sitting here on OKEX. Sitting here in pilot is Andy Lister. To my left,
On navigator is Lars Murphy, and 2 people down to my right.
Sitting co-pilot Sean Kennison.
And to his right, Bob, not on video.
And in the back in the video clipping station, I'm Emily Narrow, and we are all with the Global Foundation for Ocean Exploration.
hey Bob, what's this orange thing that's coming up?
In the water column? Oh,
Oh, here. I think we've seen a couple of these. Yeah.
yeah,
there's the
an
same.
enemy.
Okay. Come on wide.
And I'm Stephanie Bush. I work at the Smithsonian
37
Institution in
degrees.
the Department of Invertebrate Zoology.
This is, main HD camera.
What was that, Cobalt?
Maybe at 37 degrees in my camera.
Oh, 37. Copy.
Thanks for the stop, pilot. But yeah, we've seen a few of those.
Sure. And we're waiting. With ships catching up with us right now as well. So, if you want any looks at
Actually, that bright white
right away in
structure.
the distance there,
Yeah.
I'll go check it out.
I'm assuming it's going to be the shell of something dead now, but
It's a good assumption.
you never know.
Yep. Come on in. Video as I come in. We can get lasers off.
You got it.
Any ideas?
Maybe part. It's definitely part of. It's not full of anything. Part of, actually, possibly brittle star, or urchin. Not really sure. So let's keep moving because I have no clue.
Copy that. Come on out, video. There are my lasers.
I think there might be a really small
pilot
Syriantid
submits
anemone
moves.
right above it to its
About 4
left.
5.
Do you want to check that out, Scott?
No, it's fine. On the close-up you had is probably about as good as we're going to get.
Okay.
Really
Well, because
nice.
there's a large one that the lasers are about to go over. Just didn't know. Actually, yeah, pilot. Can we check out? It's right in between the lasers.
Yep. Go ahead
right
and
now.
video.
Thanks. So we've seen a few of these today, but haven't zoomed in on any of these tube-dwelling Syriantid anemones, which, when they are disturbed, the anemone can
Yeah,
retract
copy, Bruce.
into
Thank you.
a fairly long tube. So I've heard from our actual co-science lead, Cheryl Morrison. She's been present when one was
Come out,
one was
just load the
collected.
video.
Yeah, that's probably good. Thank you.
Copy that. Come wide, video.
We've been hearing about various cnidaria, of which Of course, this tube anemone is one, and their stinging
EX1806.
cells on earlier
Moving
dives,
forward.
those are structures called nidae. And these tube anemones have a special form of nidae called a tychocyst, and they will fire those structures, and they are sticky, and they use that along with mucus and the sediment grains to actually build the tube that they live in. And they're the only cnidarian that has that kind of structure, the tychocyst. And the way it's described in the textbooks is it's the thread-like part of it that's extended is folded up kind of like a fire hose that you would see in the hallway of a building, for example, in an emergency fireplace.
Yeah, I forgot about that. You're taking me back to way back when I took invertebrate zoology. Hey, pilot, just in the distance a little bit and left of center is another sea pen. If we could check that out and see if it's the same as the one that we collected before.
Absolutely. Go ahead, video.
Other than having some seaweed and mucus stuck to it, this looks similar to the one that we collected. Do you agree, Scott, or do you think this is...
I do agree. I think this is the same taxon. It looks like maybe the top of this one, the polyps have been retracted back, and so maybe that axial skeleton, the rod, is exposed. That might be what the black thing is at the very top. And yeah, it looks like
Oh, yeah.
or some other gunk, sargassum or something has got caught up on the colony.
Yeah.
Yeah, there you go. You can see the internal rod that forms the kind of like the backbone for the colony. Again, I'm really struck by the color, this purple and pink of this colony that I don't recognize. Thanks for that close-up.
Thank you, pilot. I think we're good on this one.
Okay, ready whenever you're clear.
Got multiple types of anemone over on the left. See lots of these tube anemones now, or actually for the last little bit, but we're at 1,157 meters now. It's quite a big divot in the sediment there.
That's odd.
There's no way that that's from us, right? We haven't, this is, we're like going in.
We have not made it there yet.
Okay, didn't think so, just want to check though.
But very strange. Maybe that shrimp had something to do with it. Try to get a flyby. Yeah.
Or something to your left. You want to rotate after that.
What's that?
I think I saw something vaguely
Oh yeah,
to
what?
your left.
Where are you seeing it?
Just pretty much directly if you just turn left on the series.
Oh, in the lights?
Yeah. Looks like a trail, but larger. It might be nothing.
Looks like a couple eel pouts on the right side of the screen right now, and...
Current picked up.
Yeah,
Hey,
it's nothing,
pilot.
My bad.
Go ahead.
Can this something lower right side of the screen? We're just about to pass it. I don't recognize it,
Sure.
although it's probably something we've seen. But can we zoom in on that one?
Talking about the guy that's pretty much off the screen right now?
Yeah, exactly.
Another taco,
Thanks.
isn't it?
Might be. Second Choco Taco. Let's take a closer
Yeah, and I
look
think
as
you're
I
right.
land
It's one of
here.
those cup
Whoops.
corals that folds in half.
Okay, come on in video.
Yeah, so this is another one of the cup corals that apparently the only type of cup coral that can attach to something really small so that it can survive in highly sedimented areas like this, called flabellum. That's good. Thank you, pilot.
Copy that. Where are we in moves? Are we still moving? Not moving?
We're currently stationary. We have been for the last 6 minutes.
Copy that.
So
So
for
he'll
you
be
to
coming to a...
push ahead a little bit. And then
Yeah,
we're gonna
he's
make another
probably
move.
still finishing up his swing.
A couple sea pens on the left here.
Do you want to see the sea pins or this structure here?
We could quick zoom on the sea pens. I think they're the same, like the same that we just looked at, but it might be nice to check.
Okay. Go ahead, video. Frame it up as I come in.
Yeah, it looks the same in the other few, like the one we collected and the one we just looked at, so that's good.
Copy that. Come on, wide video.
Well, Scott, did you notice on that one, the stem structure was exposed? Oh, you think it's different?
Yeah, that's absolutely different. That one has what we call polyp leaves. So the structures that were coming out basically left and right of the main axis, the polyps were arising from there. Whereas the one that we collected, the polyps come directly out of the axis. So that's new for the day.
I can hear him, so you're going to want a closer look at it?
Something different.
Okay.
So, Scott, is that one that you are familiar with, then?
Not 100%.
Come on in, video.
So we should get a really close look. So
Yeah, we should get a good look. Yep.
Okay.
And you can
we have
try
the
the
time,
fog
absolutely.
lights if you'd like. Might be a little bit of sediment though.
Try
'cause I'm
video. Great.
on the other side of it.
Fog lights coming on.
Yeah, so you notice here you've got basically the main stem, and then from either side arising is this little shelf, or what's called a polyp leaf. And then arising away from it, you can see all the individual polyps are arising from that. And this is common, for example, in the genus Panatula. I'm not saying that this is Panatula, but...
Okay. What about the, I was noticing on the other ones, actually, the pale, very pale pink round structures. Is that reproductive
Yeah,
parts?
I'm not seeing it well enough.
Would you like me to tilt up or good view
I'm
here?
not sure. I can't. I don't know if I can hear Scott anymore. If he's just not. He might be researching.
Oh, sorry. Can you hear me now?
Yeah. Yeah.
Okay, it's really weird. I was picking up my phone to move it to be better able to see the screen, and it seems like whenever I pick up the phone, I must be blocking the microphone.
Okay.
So what I said is that there are some sea pens where fish actually lay eggs on them. And I'm not saying that's what we're seeing here, but I don't have a good enough view to know if those are eggs or gonadal structures within the polyps of the sea pen themselves, or if it's something that's on top of the sea pen.
They're more numerous on the top of the colony,
I'll
if
come
that
up to the very
indicates
tip here.
anything. More numerous and or
Not to
larger.
me.
With all those particles in the water video, do you think that we're making it any
Yeah, I
worse?
mean, I don't know
with
if
the
you
fog
can
lights?
tell if they were gonads, they would be inside each polyp towards the base, or There are structures called
Yeah, let's give it a try.
siphonozoids, which
Lights
are
on.
modified
Just like
polyps,
frogs.
and they would be at the leaves,
Those
at the
don't
base
show
of the
up here as
leaves,
well, do
and that
they?
would be often where you would see
Turn
the,
back on, or-
the gametes and the gonads. So maybe that's what's going on right there.
We're trying to get those eggs.
Looks more like the latter thing that you were talking about. You think we have enough video on this
Tina's
to...
saying it's not the genus Pinatula.
Oh, okay.
So it is what used to be called subsessifloriae, meaning the polyps arise on these ridges or these leaves. But I'll have to do a little bit of research to determine what genus. And interestingly enough, as the ROV did this really nice job of sweeping around to this side of the colony, now this does look more like what we collected, and maybe I just didn't realize that the polyps were arising on leaves on the one that we got, and it's just that we had the back view when we came across this one. So that was very, very useful to see both sides. It'll help us in the future to identify these things from imagery.
Great. Sounds like we might be done here, watch lead. Oop. Shift change there. Okay. Coming up.
So is the decision to not make a collection? This is Cheryl. I just got back. Sorry I had to step away for a minute.
Yeah, the decision was not to collect that. It may be like the one we already collected earlier.
Okay.
Thank you.
Sure.
And welcome back.
Thank you.
Thank you. Thanks for helping us out while we were gone.
There's another sea fin coming up. Do we want to look at that too?
Watch lead, this is Nat.
Okay, we'll continue past this
Just
one.
to fill you in, we have moved 200 meters since we started, and we have taken one sample, which was a sea pen similar to the one that you just saw on video. That's in starboard inner.
And we've gone 200 meters since we reached the seafloor. And we are at a depth of 1,153 meters.
Copy that. Another 30 meters at 305.
Do you have eco-pilot?
Or do you want to bump it down to 20?
You can do 30. It's starting to...
Yeah, according to HiPAC, you're supposed to flatten out quite a bit, and then it will steepen up in a while.
Yeah, I think you could do 30, no
It's
problem.
like little footprints climbing up the slope there.
Lined up about 305-ish.
Copy that. So 30 meters at 305. Bridge, this is ROV now. Would like to call another move, same thing, 30 meters at 305 degrees, speed 0.2. Good copy, Bridge, thank you. Go ahead, watch one. We have not looked at either side yet. It's a pretty wide ridge.
This feature that we are visiting today is an Intercanyon Ridge.
Yeah, it's not narrow.
It is one of the submarine canyon ridges, or between sort of finger-like canyons that
moving
occur
away from the center of the ridge pretty
along the North Carolina
move a little bit further
continental
up towards the
margin,
northeast,
and actually many of these all the way up to Nova Scotia. There aren't as many of these canyon features south of here as you move into the South Carolina, Georgia, Florida region, and that's largely because of the broad Blake Plateau area that we were exploring earlier on this mission. Instead, here we are looking at canyons that have been carved through millions of years. Some of these are as big and deep as the Grand Canyon out west in the U.S. This particular one is a fairly
head off to the side
simple canyon in that it's one conduit starting at about 200 meters water depth down to the deep sea. And there are several of these all lined up along what we call the continental slope.
Over the side of this ridge.
Where we are actually diving is one of the ridges between these individual channels or submarine canyons. It's elevated relative to the channel bottom, which is another several hundred meters deeper than this. And we are slowly climbing up, it's like a backbone, slowly climbing one of the steeper portions of it in hopes to find areas where this thick sediment drape has been swept away by the Gulf Stream's currents, exposing some of the underlying rock. And in that kind of habitat, we would expect to and certainly hope to find other groups of organisms from what we're seeing on this portion of the dive, and namely looking for deep sea corals and sponges that need that hard substrate to attach to.
Probably be more like 295, 300.
This type of sediment drape that we're looking at is apparently not uncommon. Yesterday's dive was in a similar ridge setting. It was within a much larger submarine canyon, but it was still that ridge-like feature, and we climbed up its backbone and saw a very similar type of seabed. Yesterday we saw a lot more brittle stars, so at least the community of organisms is different here compared to that other location. But one of our colleagues from the U.S. Geological Survey, Jason Chater, who I don't believe is on our chat room right now, but he said that at these water depths, the Gulf Stream's current isn't quite strong enough to keep these areas free of sediment deposition, and so there is a thick sediment coating here, and that maybe on some of our upcoming dives that are shallower, we'll see more exposure of that rock. So it'll be a great comparison and provide a lot of information for us as to what kinds of substrates and therefore what kinds of habitats occur along these margins.
As far as the naming convention,
we'll be able to say after 2 dives that look like this type of seabed that they are not coral and sponge habitats. They are homes to very different groups of organisms. But perhaps in upcoming dives where we're shallower, as I said, we'll find more rock exposure and denser communities of the deep sea corals and sponges.
Starting to move a little, so you have tether to use.
But this kind of habitat is also interesting because there might be more benthic diversity besides the corals and sponges that we've been seeing a lot. So it's an important comparison, and a lot of the seafloor looks just like this soft sediment, and maybe organisms are spread apart. They're not concentrated like we see them at coral areas, but there's a lot of food here, even though it doesn't look like it necessarily, but a lot of organisms have a fine time making their homes in soft sediment conditions, and there's a lot of food within all of those particles that are falling to the seafloor.
Now, this is like the farmland of different countries where people are spread apart a little more. Homesteads are more spread apart, but there are resources there to keep them
where this is such a flat area,
healthy and protected, and there's plenty of food. It's just not quite so concentrated, and those rocky outcrop areas, everybody wants part of that, so they're all clustered very tightly. Those are sort of the cities.
All systems are go.
How's the DC bus look on Bull?
Sirius looks okay. Better than it was.
Good copy, Bridge. Thank you very much.
Let's see. D2 looks good too. Yeah, all looks good.
Would you all be comfortable with another move? It just finished. I know there's a little bit of laid back, but you're pretty far ahead.
Feel pilot?
Sure. Which direction are we going now?
I can rotate to, you said 295? Is that?
Yes, 295 would work well.
Copy.
Yep, go for it.
Copy that. Slightly port to 295.
30 meters at 295. I will call it in Now it looks like you're there, Cobot.
Yep.
Bridge, this is ROV now. Would like to make another move, 30 meters at 295 degrees, speed 0.2, please.
It's a lot of mud.
Good copy.
Mud is actually a scientific term. It's one of my favorites. And it means the combination of the two smallest size fractions of grains, the clay-sized particles and the silt-sized particles. So when you have sediment that combines those two sizes, we call it mud in the deep sea, or even in the shallow sea, but it is a truly used term. If it were all fine clay-sized particles, the microscopic, if you rub clay between your hands and feel no grit, that would be just clay. And these sediments, we took a sample yesterday. These samples are largely made of that finest grain size, which tells us that the currents aren't as strong here. You need very low velocity currents in order for those finest particles to settle out and form the seabed. The silt size, the next size fraction, silt size particles are primarily made of the shells of planktonic organisms, single-celled foraminifera.
And this looks like possibly another flabellum, is it?
Looks like it to me, Cheryl. At least some kind of cup coral or scleractinian coral in the mud.
Yeah, and we've been seeing something similar quite often on this dive, and that would be my guess, is that it's a cup coral in the genus Flabellum, because of that group of species lives in soft sediments.
I see a lot of stalk type. I don't know if they're tube worms, the tubes of tube worms. They don't look like echinoderm spines. Has anyone noticed that or addressed that? Particularly to the right of this organism. I hadn't noticed that in previous.
We had a for Leslie where we saw a lot of this stuff, but we didn't really remark on it, and I'm not sure what it is.
Kind of over here, watch
debris of some kind.
Yeah, it's interesting. We haven't noticed that before.
It's a beautiful coral. And now we can call this a coral dive, right? We've seen at least one species.
And it is deep, so it's a deep coral dive.
Mm-hmm.
And we've had both scleractinians and octocorals, so it's covered.
Clear here. Good.
And serianthid tube anemones, too. And just so everybody knows, we're going to try to move a little bit more upslope and get to the top of this and try to peek over to the south-facing ridge before we leave the bottom. It's a great shrimp. These look like bigger burrows.
Yeah, they definitely are large, aren't they?
And a rat tail, Nizumia.
Some debris that has settled down in there on the edges of it. And then a large, I don't know if we've seen any of the larger crabs today. We saw a few in the last couple dives. A front door and a back door, and maybe even a side door.
While you guys were on your live interaction, we did see at least one Chaseon.
Oh, great.
We also saw a Glyphocrangon shrimp.
I think that we actually saw that shrimp from the other room, and it was hard to keep my mind on what I was talking about instead of getting excited about the shrimp.
Yeah, I understand. I think that's the first one of the expedition.
Yeah, that's great.
6:30.
So, yeah.
So as you look in the edges of the entrance there, you can see a little bit darker sediment, and that might indicate that it has a little more of the clay minerals in it. And I'm curious because this mound has 3 entrances, and it may be one cavity or a network of tunnels underneath it, but there certainly has to be some sort of cohesive nature to the sediments underlying what we're seeing on the surface in order for that to be held up without collapsing. And that just makes sense. As sediments pile up, it squeezes out the water underneath and allows that material to be stiffer.
All right. Come on out.
And the clay minerals themselves are very tiny sheet-like minerals, and they have slight charges on each side of the mineral itself, and those, because of their sheet-like nature, when they get compressed, as with sediment loading on top of them, they squeezes out the water a little bit, and the slight charges on each side of the sheets allow those particles to sort of adhere to each other. They don't form a solid bond. They can be broken down easily, but just a slight attraction on each side. So the compaction being sheets plus that slight charge on each side makes clay minerals much harder to erode, actually, and it allows these sediments to be stiffer, and that's part of the reason why these sediments can be on such steep slopes. If they were individual grains, they would tumble down with gravity and also with the currents, but the clay minerals are really likely holding a lot of this intact.
Any interest here, watch leads?
Yeah, let's take a quick, sorry, we thought we were heading that way, so. Looks like he's a little wrapped up here.
Go ahead, video.
Do you want fog lights at all?
Yeah.
Video
interesting arrangement. He's got one of his arms flopped over. Right. This looks like what we have seen earlier. I'm trying to find the name of it. I certainly haven't memorized these.
I think it was Paxillosida.
That's me looking
was
at
the order.
5.
Yeah, that's what I have too.
Well, that was for the mud star. I don't think this is the mud star. I think this is the neomorphaster that we've seen on other days.
We were just testing you, Leslie. See, you really are the sea star expert.
Oh, yes, until somebody else comes on who knows more, which is very easy. So that looks, yeah, so you haven't seen this today while we were doing our live interaction?
No, we haven't.
Okay.
Very, very flexible star. That's
yeah, surprisingly. Doing yoga.
Watch these. Let me know when you've got what you need here. So
that's great. Thanks, pilot.
Okay.
Great.
One of the officers on board today, or the other day, suggested that we have a, what does she call it? Animal yoga?
Deep sea animal yoga.
Deep sea animal yoga.
We would strike poses that reminded us of some of our favorite creatures that we've seen so far on the cruise.
So that will have to go into the menu of items that we work on.
And Nolan Barrett suggested that that was neomorphaster that we were just looking at, the sea star.
you guys want to get another one in.
So we had a question from
30 meters at 295.
Sure, from Colleen John. She asked, with no rock structures or soft sediment, how are the majority of these fishes that we're seeing protecting their eggs? Do they bury them, set them adrift, or travel further for a more secure structure or cluster of seabed or rock? And the first thing that came to my mind were skate egg cases. And in the Gulf of Mexico, we've found skate egg cases very carefully wrapped around octocorals. And so they are basically off the seafloor and kind of protected by the Cnidarian octocoral.
There you go.
And then generally, it is probably more likely that most fish will set them afloat, as Colleen said. And some of them even are buoyant and will go all the way to the surface and hatch there where there's more food and then come and settle back down to the deep as they get older. But with so many species, there's probably a lot of variation as to what they do with their eggs.
Thanks for helping us out, Tammy. That was from some of that answer came from Tammy Frank.
And the reproduction of a lot of these species that we see down here is pretty much unknown, especially for the corals.
Another sea pen here.
Corals would have larvae that are going to spend time in the water column. And how much time and where they go is probably very variable by species.
At bats, you at about 3-5 degrees.
Copy 3-5.
Should be moving soon, if not already.
It's been taking about 3 to 4 minutes for you to feel those moves, I believe.
Copy that.
That was put in at 2040, so it should start to swing.
A few minutes ago, you saw that crazy hermit crab with all those anemones on it.
Come on in, video.
I know you've seen, we were very distracted doing our live interaction. We saw that scurrying across the screen several times.
We've seen
yeah,
this
that definitely
times.
looks like flabellum to me. So one of the things that we've been enjoying on the ship is Choco Taco ice cream. And I just can't help but notice that this is a taco shape, just like our ice creams.
Yep, whenever you're clear, video.
And it is Taco Tuesday.
Oh, that's right.
Is it Tuesday? Am I right?
Tuesday?
It's Tuesday, right?
You're getting tacos for lunch.
We lose track out here. Everyone had to check.
Oh, and Tammy Frank said that this is a heterocarpus shrimp. Another crangonic.
Go ahead, watch one. Copy that. We have about an hour left on bottom. No problem.
1 hour. Okay, how far do we have to go to get to there? Never mind.
We are now at 1,138 meter water depth, and we have about 1 hour left on the bottom of today's dive.
Yeah, our late start due to not being able to dive in our originally planned location, which was well offshore and much deeper water. We had a dive plan for 3,400 meters today, but the sea surface was too rough, too much wind, too much swell, too much current, and everything was in conflict with one another, so the ship could not maintain position adequately if we were to dive. So that location was traded in, and we moved into shore to this site. And so our mapping team had to find a great spot to
start going
within
west.
a certain range of distance so that we could still get in a dive today. And fortunately, we were extended our time a little bit.
Okay.
We were able to do that.
To the point area interest. So it would take us 68 minutes to get there. We have 7
Our
minutes
current
left.
depth is about 1,138 meters. We've been climbing the whole dive.
It's at what average speed?
Decimal 2.
Oh, so we'd have to.
Some of these slopes have been more than 40 degrees.
So
but like I said, we should hit this area where we can look over the side.
Where 0 degrees would be flat and 90 degrees would be vertical. So give you a sense of the steepness here. We're trying to make our way up to the crest of this ridge, this finger-like ridge, and then peek over into the side of the ridge that is facing into the current. We would have loved to have started at the base of that side of the canyon wall and climb it, but with the currents, it's very difficult for the ROV to have too much current behind it. It pushes them into the sediments, into the wall that we're climbing. So we have to choose different pathways based on certain capabilities.
Sitting there at 37. You should have another like 2 minutes.
Always keeping in mind the safety issues.
In between, so we don't
That's
stack
another
them.
very elaborate burrow.
But as long as you do that,
Certainly is. This is
keep them going as long as pilots.
whole areas.
Yeah, I'm fine.
Despite the sediment cover that is very similar to what we saw yesterday and comparable slopes, there's very different burrows and different organisms.
I can't help but wonder if that's not the giant isopod, Bathynomus, who made
that.
that.
Wouldn't that be neat?
That would be great if we could see one of those.
It's sort of shaped like a giant isopod.
It is. It's perfect shape for it, so.
And you're off to my port.
And this is the type of substrate they like to live in, right?
Mm-hmm.
That's a copy.
And when you say giant, about how large are they, Cheryl?
Oh, about a foot long.
Oh, that's what I thought.
So if you remember any of the isopods that we've seen so far, they're usually a couple centimeters, maybe. Maybe up to, yeah.
What is this? Go ahead, video.
Oh, some debris.
We found some chocolate.
That's too
Chocolate
bad.
latte.
Chocolate. Yep.
I think those are those cookies, right?
Multi-grain.
Something smooth chocolate.
Every dive.
Multi-grain.
Yep.
Yeah, I think so.
When you think of the really tiny amount of the seafloor we're able to cover in one dive, less than a kilometer in length, and the field of view, what you're looking at now, and we have seen marine debris, human-generated marine debris on every dive. So one piece of debris may not seem like a lot, but it is when you think of how large this ocean is, and yet we see it everywhere we go. We were diving on a 19th century wreck several days ago, and right in the middle of it were some plastic cups. It was so sad to see that.
And a 5-gallon plastic bucket, which obviously didn't come from that time frame.
Yeah, was that yesterday or 2 days ago? I don't. It's all running into one blur. Yeah, we've seen
good copy, Bridge.
What looked like an engine, rope, metal pieces. Something that looked like plastic wrap a few days ago.
And Tammy Frank corrected me that the giant isopod can get up to 18 inches long, not 12 inches long. And there is some literature that talks about their burrowing. And they can change the length of the burrow in response to changes in the environment.
It's kind of a ledge over there on the left. Pilot, could you swing just to the left, see if there are any burrows in that sort of ledge right in the center?
Sure.
Almost looks scoured out.
I'm pretty far, so I don't know how much tether I have.
You still have a little bit.
Great.
I'll tell you if it gets tight, or before it gets tight.
There's a pit, more than a ledge.
Yeah, if we keep an average of 0.1, we should be able to make it to the potential peaking point to be able to see over the side. So we have plenty of time.
See some traces of organisms that have been migrating across that mound. A lot of them. And there's something coming into center above.
Above or below?
Is that the can we just saw, or are those eyes of something?
Something shiny.
Yeah.
Watch these where?
Looks like a dumpling squid.
Oh, right
just
there.
above,
okay.
yeah.
That's the original.
I was hoping it might be a cephalopod's eyes that we were shining on, but
maybe it is.
It is.
Good eye.
I found one every day
Sure,
so far. It's amazing.
Another one of the bobtail squids.
Bobtail.
Video
They're so cute.
come out while I
Repeat
land.
that. Sorry, I had you muted.
I'm going to kill your lasers.
Yeah, thanks.
Oh, maybe we'll see him move. We rarely see his tentacles because he sits still. He doesn't seem very concerned with us every time we see one of these.
All right, video. Yeah, now you can come in. Oh, he's thinking about it.
Looks like he's getting ready to bolt. There he goes.
Oh,
yes.
Nice.
Cool.
And that's what they look like when they swim.
Yes.
Those are them. Okay.
They are adorable.
Come back in video.
No. They are. How could they not be favorite?
I found one of these every day that I've been pilot.
Yeah. Oh, that's such a great video.
We're going to get Stephanie online with us. Her nickname is Stephanopoulos. What's your nickname? Cephalopod.
That was a good example of the squid using the funnel.
Cephalopod.
Under the,
yes.
I'll call it the face, but basically squeezing the mantle cavity and shooting out a jet of water so that it moved backwards so quickly.
Here it goes again.
Yeah, you can see how they can. I don't just got on the line, so I don't know what was said before this. But you can see they totally tuck the fins around the body until they need to use them. And one common thing when they swim is to go pale like that. So they can change their skin color in a fraction of a second, actually.
With little chromatophores, right?
The chromatophore organs, yeah, they're just little elastic sacs of pigment in their skin, and they're surrounded by radial muscles. They retract the muscles, pulls the pigment out, and then that's when you see the color. But yeah, we got good enough view of them. You can see the individual organs on there.
He's keeping an eye on us. It's a good thing.
How did they get the name Bobtail? I don't get it.
Actually, I have no idea. You don't want to get me started about common names.
I know.
Well, then there's the dumpling squid, another common name, right?
Right.
That I can identify with.
It's pretty obvious.
Mm-hmm. Especially because most of the time we see him, his tentacles are completely tucked beneath him.
Arms.
Sorry. I'm being educated.
Good job, Brett. Thank
Subtly.
you very much.
Why are they arms and not tentacles?
They only have one pair of tentacles, the two things that come out. So the tentacles, yes, are also tucked, but you don't even see them when they're swimming.
That's what I meant.
Okay, then you could say tentacles and arms.
Okay, next time. I'm writing this down.
Yeah, the tentacles only come out when they see something tasty to eat, and they go.
We need that video too.
Yeah.
Okay.
If only we could dangle a shrimp in front of this thing's face, maybe we could get it to
That
show
would be
you.
Great. Well, I'm glad we got to see them swim finally.
Yeah, that's pretty cool.
So what should
wonderful.
should we come off of him now, or anything more we want with him?
Thank you, pilot. That was great.
All right, pilot, I'm looking at you straight down, 90.
I'm so happy that was not a can.
Me too. Yeah.
Uh-huh.
We were hopeful.
Ship is currently stationary.
Thanks,
Copy.
Cephalopod.
Watch lead descends now. Just want to let you know that we have passed 300 meters.
And we're just passing 300 meters on our track today on this Intercanyon Ridge north of Pamlico Canyon, south of Hatteras Canyon.
Can I get my lasers back on, pilot?
You got it.
Pilot.
Well, if those burrows are made by the giant isopod, there are several of them out here.
There's
Yeah.
something in this one too.
So
Do we want
really
to take
curious.
a look? Watch these.
Wish we could discover who lives there.
Oh, okay. Same thing. Copy that.
And pilot, I have about 4 or 5
The
in my
pile up of the
camera.
mound and the mounding, though, just looks more like
Yeah,
somebody has
30-meter
been
move.
digging
Definitely.
and pushing it out like a crab.
Bridge, this
But-
is RV nav.
I've never seen an isopod dig a burrow, so I don't
We'd
know
like
if
to
they
make
do
another
that.
move.
have
Same
not
exact
either.
thing. 30 meters at 295
Can you
degrees.
tell what this
Speed
is, video?
decimal 2, please.
Go ahead. We'll see if we need to get a closer look here. Yeah.
Good copy. Thank you.
Is that a nudibranch or something? It looks like the gastropod foot that we saw earlier.
There's it on a stalk.
It is a stalk, yeah.
Is it a sponge?
It's moving. Ever so slightly.
I'll bet that's the tongue of a Echiuran spoonworm
Oh.
appearing into a hole, right?
Yep, it does
Yeah.
go into a hole.
It's a tongue. That's gross.
The proboscis, or what's colloquially referred to as a tongue.
Wow. I didn't realize they were so clear. You can see right through it.
Yeah, I don't know that I've seen one that looks like this, but I'm not sure what else it would be. I mean, that's kind of an educated guess. These spoonworms dig burrows, and they typically remain in the burrow, but they can extend the proboscis for quite some distance over the bottom.
Oh, is this the one that you talked about several dives ago, and you could see almost the radial pattern of where it had extended?
Exactly.
Okay.
Yeah, come on that video.